Thanks for having me. I'm a research fellow here at CCB. I've been here for just a year, a little over a year now, and I'm going to tell you about two projects that I've started while I've been here, some results from those projects, and some large scientific questions that we're hoping to answer. And when I got here, I was a physicist, but somehow I've turned into an entomologist, which was a bit of a surprise. So this is a conference on algorithms, and if you want to find algorithms, developmental biology is a great place to look. So you'll see, for instance, when one cell splits into two cells, that's an algorithm from 1 to 2. If you have a bunch of neurons arranged and they form connections to join their neighbors to form nerve fibers, that's an algorithm. And if you have, this is a picture of a Drosophila embryo, and this is a cross section of this round ovuloid, and there's a process where some cells at the bottom bump up and transform to be on the inside of the embryo. This is a very important step in morphogenesis. It's also an algorithm. So at these 3 different stages, from single cell to multiple cell to entire organisms, algorithms govern biology, and the hallmarks of these algorithms is that first, there's self-organization. So there's no top-down view telling all the cells what they should do. They communicate with their neighbors and self-organize to form these structures. Reproducibility and robustness. So every adult fruit fly started as an embryo that looks like that and had to go through this step to make it into adulthood, and ordering independence of steps. So you start at one step and then something else happens and that step has to finish before the next step can take over. So I'm going to split algorithms into these 2 types based on the features of their outcomes. So if you look at the school of fish, you can see 2 types of algorithms. First are deterministic. So these are algorithms that generate the same structure every time. So in all of these fish, you'll see 2 eyes, 2 fins, and 1 tail. So unless something went really wrong genetically, you'll have all these features in the exact same places. But if you look at, for instance, all of the dots on these fish, you're not going to be able to find 2 fish with the exact same pattern of dots. So these are algorithms that have generated variable features that are unique to each fish. And I'm actually going to study something that lies in the middle of these 2 algorithms. So something that has both the deterministic and a stochastic component. These are two examples of these types of algorithms. So if you look at the wings of a dragonfly, of an adult, or the tracheal system of a fruit fly larva, what you'll see is some things that are the same and are always the same, found in every single animal, and some things that are unique and actually kind of serve as fingerprints to an animal. So these are not just two special examples that I've looked for and came up. They occur over and over again in different places at lots of different length scales. So it's important to understand why nature selects these algorithms that do some things deterministically and other things stochastically. So the roadmap of my talk is, first I'm going to show you these 2 data sets of similar yet non-identical features. I'm going to explain graph methods that we use to analyze these networks. And in the end, we'd like to quantify variability using the left-right asymmetry. So a close-up of my first example, these are these are the 4 wings of the adult dragonfly. And what you can see, you can pick out already, are these 2 different types of features. So the primary veins are these long longitudinal veins that start on the shoulder and go all the way to the wing edge. And you can pick them out by eye, and they'll be the same for every specimen in a given family. And we can take our image and convert it to a network. So these are no longer pixels on an image, these are nodes and edges. So I can talk about G1, which is the top wing, and G2, which is the other side. And I've given you a size of what these look like. So they have around 1,000 nodes and 2,000 edges. My data set will be 11 dragonflies from the same family.
Tatyana, so are the connections between the non-primary veins, are they also veins? Or what makes a connection?
They are veins.
Okay.
I mean, they are not hollow, so they're connections. So they're tubes, and then in the middle there's a membrane. Okay, so we're talking about asymmetry. Let's think about the most basic measures that we can look at. So comparing left and right sides, so red to red and blue to blue. Or we can talk about just the area that a wing takes up. And you see, okay, the forewings are indeed smaller than the hind wings, but let's talk about the asymmetry. So this is % difference in area between the forewings and the hind wings. So the forewings, there's less than a 1% difference, and the hind wings, there's about a 2% difference. So you can think about this as comparing the length of your arms, maybe a 1 to 2% difference, you wouldn't really be able to tell. The other thing that we can compare is the number of faces in the wing, and so the forewing is smaller and has fewer faces, but about 700 faces in both of these wings. And again, there's not that much of a difference. So there are differences, and this will be a problem when you're trying to match a graph of one size to a graph of a different size. But the point I'm trying to make here is that it's still pretty symmetric. So from these basic measurements, the levels of symmetry are quite high. And let's look at the other components of these wings that are deterministic. So I'm going to define a cable as a path of edges. And in these networks, I'm going to find cables that are longer than a given threshold, longer than 10 steps. So if I draw them for all 11 wings and place them on top of each other and match the boundaries to each other, then you'll see these long cables that turn up in all the wings, and these are going to be the primary veins, the deterministic components. And I haven't done this yet, but you'll have to believe me that this picture identifies all of the primary veins in these two wings, and you can see there's two types. So there's the red types that start at the shoulder and continue on, and then there are also these mysterious blue types that kind of start in the middle of the wing blade and then continue to the tip. Okay, so these are the deterministic components. Let's move on to the stochastic components. And I want to find where in the wing things are different and how different they are. So how different they are and how that varies spatially. So what I'm going to do is solve this graph alignment problem. So the graph alignment problem is if I want to map one graph to another graph, see where things are the same. This has been done in protein-protein interaction networks, where these are large networks in high-dimensional abstract spaces, and the edges are measures of similarity between the nodes. So there are some examples of algorithms to solve this problem already. And the way that the problem is set up is you're looking for a mapping, which is a set of node pairs where the first node is in one graph and the second node is another graph. So you can think of lots of mappings here. Really, it's just a combinatorial problem to find all the mappings. So how do you say if one mapping is preferred over the other one? Well, I'll say that each mapping comes with its own set CM, and that's the number of conserved edges. So this is just, I'm looking at the number of edges that one edge is in the first graph, the other edge is in the other graph. And then if you look at the node pairs that make up that edge, you'll find them in this mapping M. So the goal is a combinatorial problem to find the mapping M with the largest CM. So the thing that we can do here that the protein-protein interaction people can't do is that we have geometric information. So if we align one wing just on top of each other, matching the boundaries, then you'll get pretty pretty close alignment in some places. So for each node in the first graph, we can construct a candidate list of nodes in the second graph and know that in the mapping, this node needs to be mapped to one of those. And already this has reduced the combinatorics significantly. So in a wing of about 1,500 nodes, if you have unrestricted alignment, so any node can map to any other node, that's 15 factorial, so that's about 10 to the 4,000 combinations. But once you have this candidate list and do this geometric restriction, you only have okay, you still have a lot of combinations to check, but it's become a little more tractable. So the way I do this is I start at a guess configuration and then do a Monte Carlo assignment and switch between different but related mappings. And the way I pick whether to stick to my mapping or to go to a new mapping is evaluated by the set CM. And the way I narrow things down is I use these crystallized faces. So if I have this set of nodes in the first graph and I've mapped them to the set of nodes in the other graph in my mapping, then I say that, okay, obviously the only choice for N1 is M1, so I fix the candidate list of N1 to just be that one node, and I remove M1 from all the other candidate lists. So that kind of narrows things down and I can build up my set of matching, my matching set. And then I do one more thing. So I initially run the alignment on the unaltered graphs, but then I'll have a lot of cases like this, where two nodes, N1 and N2, or where N1 is mapped to M2, but you have this topological mismatch. And it's clear that if you just make a topological transition and combine these nodes into here, then you'll get a better alignment for the other edges in the set. So what I do is align it once and get all the green faces that have been crystallized. Then I do this set of topological shifting, and then I run it again and get this next set of gray edges. So this is what it looks like for a single animal, the left and right wing. So first I can fix the right wing and align left to it, or vice versa. And I'll get about 18% face alignment. The other way I'll get about 26 face alignment. And you can see there's a distribution of these green places where it's an exact match, and also places where it's a near perfect match. So this is an aggregate result of all full and partial alignment across my entire data set. On average, it's about 21% alignment. And what I want you to see in this picture is that it's not a uniform orange color across the whole vein. The colors serve as a heat map of alignment. So where the color is strong, that means there's a lot of alignment there. And when the color is weak, that means nothing has aligned in that region. And what I want to do now is to correlate regions of alignment with other features of the network. For instance, where these main veins form. So what I've shown are not strictly the main veins, they're main veins plus extra stuff, but definitely all the main veins are captured here. And you can see, you can kind of maybe convince yourself that the main veins correlate with regions of color, and I'm working on quantifying that further. But another thing, another observation that seems to be messing up the alignment are these degree 4 vertices. So these wings, most of the vertices are degree 3, so 3 vertices matching at a node, but some of them are degree 4. And if I plot all of my degree 4 vertices on the left and the right wing on top of my alignment, then I'll kind of see that the X's occur in the white spaces. So you'll have degree 4 vertices in places where there's no alignment, and places where there's good alignment, the boundaries almost seem kind of pinned by these degree 4 vertices. So my hypothesis is that these vertices are somehow impeding alignment. And These vertices are kind of odd to see in a biological network, for instance, in vasculature, in plants or animals, or in foams where the dynamics are governed by line tension. You wouldn't see this kind of thing. This would be energetically unfavorable. So it's kind of confusing that these things are showing up, and understanding the origin of these types of vertices will give insight to the developmental algorithm of the wing. All right, so that's the end of this first part. Now I'm going to move on to my second example. So this is the Drosophila trachea. So this is a larva of a fruit fly, and this is its tracheal system, so its respiratory system, how it gets oxygen and delivers it to all the cells in its body. So the way it works is that there's 4 openings, 2 at the head, 2 at the tail, and they lead into this plumbing of internal pipes that link together and match up, and the oxygen is just enters at the spiracles, the openings, and then just diffuses freely and is able to be distributed. But what I'm showing here is just the cartoon. It's not the full structure. What's missing are these finer structures, and they're called terminal cells. This is actually where oxygen exchange happens. So when you're in these larger tubes, oxygen does not leave the tissue. It only leaves the tissue at these finer cells. Yeah?
This is an unfolded skeleton system?
So this is
1 centimeter.
1 centimeter. It's about 5 centimeters long.
Right, so this is some sort of folded thing?
Can you use your seat mic, please?
No, it's not. So this is the top-down view. So the scale bar is right. This thing is about 5 centimeters, 5 to 6 centimeters long.
Millimeters, maybe?
Is that what I mean? It's not that big. This should be millimeters. Right, okay. Sorry, I changed the length scale. It was 0.5 millimeters. I doubled it, so it's 1 millimeter long. Yeah.
These air tubes look like they would be good for pressure-driven flow to pass through them. So does the thing breathe? Is there a net flow through this, or is it just diffusion?
So the thing breathes, so it consumes oxygen, but there's no lung structure. Older insects do have air sacs that provide a pressure difference, but
Okay.
This is purely diffusion driven.
Wow, impressive.
Right. So if you zoom in on 2 of these nubs sticking out of here, then you'll get this whole feature. So this is the left half is this nub and the right half is this other nub. And what's shown here are only 2 cells. I'm pointing out the nuclei right here. And so they don't look like regular cells. They have these long, intricately branched structures. And we want to understand the formation of these structures. So the goal of this project is to compile a data set of hundreds of these terminal cells and study the statistics, the general statistics of them, which has not been studied to our knowledge. So there's quite a bit of variation over these terminal cells, but maybe if you think about it, you can tease out some common morphology. So this would be analogous to the primary veins, to the primary veins of the wing. So we're looking for structures that are present in all of these, in all of the cells, and we'll call this morphological backbone. So the first question is, does it even exist? The second question is, how do we find it using graph alignment methods? and something similar that we used for the networks, we want to use for these networks, which are tree-like. And the difference here is that before, where the most important factor was really the topology, here the geometry comes into play as well. So these are the channel radii, the lengths, the branch angles, and all of this will need to be taken account when aligning the left half to the right half to find the deterministic features and the stochastic features. So future work of these projects is to map structure to the function. So the function of the wing is, of course, to fly, and the role that the secondary cells play, secondary veins play in the role of this function is, well, if there's too many of them, then they're going to be heavy and mess with the agility so you need to be light to be able to fly. But also the purpose that the secondary mesh serves is resilience to damage. So if you have a crack in the wing, then it's going to be secluded to a particular region, and it's not going to spread and cause catastrophic destruction in the whole wing. So with this fine mesh, you're kind of weighing weight versus robustness, and that's something that the wing has to figure out how to balance and optimize. And the function of the trachea is much more simple. It's oxygen delivery. So it needs to, every single cell in the trachea, in the fruit fly needs to be within several microns, say, of one of the tracheal branches. And the way the system works is that oxygen enters at the root of this tree, and then as you're going along the channel, the walls are leaking oxygen, and it's leaving and feeding the cells. So you have this type of perfusive system, and what we'd like to do is to map each structure like this to the total amount of oxygen that it provides to the animal. So obviously we have a distribution of structures of these geometrical trees, and we'd like to see, okay, how much difference, how much variability is there actually in the functional space? So if two of these terminal cells that look different can actually deliver the same amount of oxygen to the animal, then that's a reason why this stochastic algorithm is still providing good functionality. All right, well, so the summary of my talk is I've presented these two examples, and they're examples of similar but non-identical structures. And the idea is that studying them gives insights to mechanisms controlling development. And especially this left-right asymmetry is important because when you study this algorithm, you want to identify where the stochasticity arises. So is it the algorithm that's actually stochastic, or is it because it's developing in a noisy environment and there's stuff getting in the way of it doing the same thing over and over again? So we'd like to, because these came from the same animal, both of these structures are first genetically identical, and second, they've developed in identical developmental conditions. So studying the left-right asymmetry actually indicates leeway that's built into the algorithm. And then finally, we like to map these to their functionalities and study the interplay between structure and function. So I'd like to thank Lena, who does all of the experimental imaging in the tracheal system and has imaged and traced hundreds of these terminal cells by now. I'd like to thank Jody Schoenfield-Romes, who's an expert in Drosophila trachea and has been a very good advisor on this project. And thanks to our very own Aaron Waters who has helped with the 3D visualization of the tracheal cells. And thanks to the Schwarzman lab, of course.
Any questions?
Sure. Very nice, very inspiring questions and data and talk. Have you tried mapping a left wing of one animal, one individual, to the right wing of the other of a different individual? It seems like if you're trying to get at how much is to do with environmental noise and how much is to do with sort of noise within the same animal, then you sort of compare whether the left wing can predict the right wing of the same animal better than if the left wing of a different animal can predict the right wing of a different animal, right?
Exactly. So that's definitely in the works, and I want to come up with statistical measures that that speak about aligning the same animal to itself versus the same different animals. And I think that that'll be a good probe of the actual algorithmic noise compared to developmental noise.
Right. Yeah.
So in the works.
Okay, sounds good. Yeah, I mean, sounds like there's some ideas from causation in statistics, or whether one measure of if A causes B is whether if you have the data from A, you can predict B better than if you didn't have the data from A. So you just apply that in this case. So anyway, it sounds fun.
Yeah, very rich experimental system.
Okay, Mike, you had a question? Okay.
Then I have, since you're now an entomologist, I didn't understand something about the physiology here. So at the level of spiracles, those things are air tubes, right? And at the level of those cells, the oxygen is those things are now fluid. I mean, that's actually that's intracellular stuff over there, right? Those are not. It doesn't have tubes.
Okay, so the physiology of this is that these are hollow tubes.
Those are hollow tubes.
Yeah.
Oh my God.
So there's a hollow layer.
That's a single.
Inside.
There's a cell that forms branches like that. That each branch itself is a hollow tube.
Yeah, I don't have I got rid of the cross section, but yes, each branch is a hollow tube, and we can image the inside of the tube. So it says airway surrounded by cytoplasm, surrounded by the tissue, and we can image the radius of the airway, and it actually varies. So if you're down here in the bottom of the cell, it's going to be the difference between the inner radius and the outer radius is going to be quite large, and then later on it's going to vary, and that we think will change the rate of how fast oxygen is able to leak out and go into the other cells. Yeah?
Yeah, I got it. That is frightening. They don't make people that way.
So is this like 100 microns across in radius?
Yes, roughly. So where's yeah, a few hundred microns.
It seems like a nice PDE problem to solve, which is the diffusion of oxygen through these airways and into the surrounding liquid.
There certainly is, and that is very much in the works.
Okay.
So that's what we want to do, and map each structure like this to a number. So how much oxygen is this able to deliver per second?
Sure.
Are you sure? How definite are you that it's purely diffusive? Because I assume all of this is surrounded by muscles that are moving around and things like that. There could be some peristaltic part on large scales. Peristaltic?
So there's no flow.
Are you sure? How do you know that?
How do we know that? So
you in the field, but we will be happy to challenge it.
Okay.
And I think that the calculations of the flux are actually necessary to know whether this thing can do the job of feeding as much surrounding mass as it does.
Does this work? Yeah. Yeah, but I still got this here. So, I actually have two questions, but I'll just ask one. And you have the right slide up here. So there's basically these black blobs at the end of the wings, which seem which seem to be something that's very systematic. And I was just wondering whether you could use this in any way.
For alignment?
Yeah.
Sure, but there's only one of them per wing, right?
Right, but
so the black blobs, they're called pterostigma, and they serve as a counterweight. They're very important for acceleration in the wing.
Okay, cool.
and sure, you can use them. I mean, the alignment is pretty good at the wing tips already. So really, it's here that I need help with.
Hey, oh, still someone's. Sorry. Yeah, go ahead.
Oh, okay. Well, thank you. Well, very cool talk. And a follow-up question on Alex's question, and also because Mahilu's not here, I'll ask her question that she would ask. If you, in the work you have in the pipeline of comparing the wings, because they have different numbers of cells and everything, have you considered using a sort of transport map, like an optimal transport map, to find out how far the left wing is from the right wing? Because otherwise, that would give you a lot of structural information of how different they are.
Right. So I've looked at these. There's this topological Earth moving method to transform
exactly
one
that.
network into another.
Earth's move resistance is exactly that. That's the optimal transport map.
Will that give me spatial difference of where the things are located? Because what I really want is this heat map of hotspots of where things are well aligned, where they are not aligned. So give me like a number. So how many moves I need to make to get from one wing to the other. I want the spatial resolution.
I think you could. Yeah, you'd get an entire map of how to move every single, like if you think of the nodes of that graph, you'd get the map that would move every single node in sort of the mass of that node into the other nodes on the other wing. So maybe something like that. Yeah, I think so.
So Robert, do those algorithms for like discrete Wasserstein distance, the kind of Earth moving thing, don't they introduce mapping?
Same kind of, what do you mean exactly by same kind of combinatorics?
Well, I mean, here it's fundamentally a
Use your seat mic, please.
problem. A gazillion things on one side, the other side. There's a lot of ways to map them together.
It doesn't, I mean, it doesn't consider, it's not combinatorial in nature. It doesn't consider all combinations or go through all combinatorial things. And I mean, I think the original smoothest distance does, and that's why it's intractable. Sorry, it does actually. So the original one does. But nowadays, everyone uses a regularized version with the, what's the algorithm called? Synchron algorithm, which is a regularized version that doesn't do all the combinations.
I would just take one more question. There is a reception after this here. And then maybe other questions you can take offline.
Thank you for the talk. I was just wondering, probably a naive question, but you mentioned that like degree 4 vertices are what's kind of messing up your alignment. Is alignment better if you kind of artificially break it up into 2 degree 3 vertices to make everything uniform degree 3? Is that, you know?
Right. Well, so because there's 2 ways to make 2 degree 3s that are degree 4, it's easier to collapse everything to degree 4, right? Or where it occurs. So that's what I do partially here. So that's the difference between these green and the gray regions. But yeah, something I'm looking at too.